Are you working for something exciting that you really care about? If yes, so you don't have to be pushed. The vision itself will push you. So just execute, stop talking about what you are going to do and start doing with it. Put into your work and let the results do the talking. So hey friends, I'm Riya Khilwani from Biotechnology. Today I will be talking about GATE Life Sciences syllabus hacks. For your information, I have come up with the important topics for all the sections that are there in the GATE Life Sciences syllabus that you can also find a link below. So you can just visit the description box for this, download the syllabus and start preparing for the upcoming GATE exams. So with no more delay, let's proceed with this video. So the first question that comes is, what is the advantage of giving GATE Excel while you are preparing for CSIR NET Life Sciences exams? So if you have seen the PYQs of GATE as well as CSIR, you will find there are two core sections in CSIR syllabus. There are section B and section C. Section B simply talks about the simple conceptual questions on a subject, whereas section C, it deals about the experimental based questions and also research oriented questions on a topic. So in comparison to CSIR, if we see GATE Excel exam, it only talks about simple conceptual questions. You will also find some application based question, but the difficulty level is simple if we compare the part C of CSIR NET Life Sciences exam. So if you are preparing for CSIR NET Life Sciences exam, you are a step ahead. You're automatically preparing for other exams too. So you can see how advantageous it is. If you are preparing for CSIR, you can write other exams too. So at once you are hitting more than 2 targets. So now let us see what is the GATE Excel exam pattern. So if you see, the exam duration is for 3 hours, that is 180 minutes. You will get 3 hours to solve your question paper. What is the exam mode? It is CBT. That means you need to be accustomed to the online mode of examination. Total question that is asked is 65 and the total marks for the whole paper is 100 marks. So what are the sections in the GATE Life Sciences syllabus? You will find there are 3 sections under this syllabus. The part 1 is general aptitude. This part is compulsory for all candidates. It further includes verbal ability as well as numerical ability. In part 2, you will get chemistry. Again, this is the most scoring section and compulsory section for you all. You should attempt all the questions that are there in this chemistry part. Part 3 deals about the subjective parts. You will find there are 5 sections under part 3. First section is biochemistry, second is botany, third is microbiology, fourth is zoology, and fifth is food tech. You can select any 2 subjects of your choice and attempt this. So, and for this 5 sections, you just have to select 2 sections out of this and attempt your syllabus. So now let us see what are the type of questions that are asked under GATE Life Sciences syllabus. So you will find 3 types of questions that are asked. First is MCQs, that is multiple choice questions, where you have to select a single and suitable options out of 4. The second pattern is multiple select questions. There you will be getting a question and you will have 3 to 4 options, and out of this you have to select more than 1 options. That is what called multiple select questions. And the third type is numerical answer type, that is numerical ability questions, where you have to solve the given numericals and put the answer for this in the space given below. So now let us see what is the design of questions that is asked. So the questions are made in such a way to test the logical reasoning of the candidate. You will find an application based question in a GATE syllabus. You can also find the analytical question that tests your analytical ability, and also you will find a comprehension based question which is going to test the comprehensive skills of a candidate. So you will find 3 types of designs of questions under the GATE exam Life Sciences. So now let us see what is the marks distribution. So I've already mentioned earlier, you will have 3 sections. First is general aptitude, that is compulsory for you all. The second section is chemistry, that is again compulsory, and the third section over here is 5 section, out of which you have to select only 2. All right, so it depends on your interest which sections you are going to attempt in the part 3rd. So if you see the type of question, you will only find 1 marks and 2 marks question under GATE exam. So in general aptitude section, you will find 5 1 marks questions. For this, that means 5 into 1 is 5. So total 5 marks questions for 5 questions for 1 marks, and 2 marks questions you will again find it 5. So total it is 5 in number. So 5 into 2 is 10. So general aptitude is asked for total 15 marks. So if you will include 5 and 10, it will come to the total of 15. So total of 15 marks questions are asked from general aptitude itself. So it covers 15% of the syllabus. Now coming to chemistry section, you will find 1 marks question. Again, it is same as in case of you saw in case of general aptitude. You have 5 questions for 1 marks, that means 5 into 1 is 5, and now you will have 10 questions of 2 marks. That means this time 10 questions you will have for 2 marks, that means total 20. So now if we count 5 and 20, it will be total 25. Now you can see this chemistry constitute 25% of the total syllabus. Now coming to part 3, that is the most scoring and important subjective part. So under part 3, you will have 5 sections, or out of which you have to select 2 sections of your interest. So select accordingly. So here you will get 10 questions of 1 marks. So 10 into 1 is 10 itself, and you will also get 10 questions for 2 marks, that is 20. So now you can see 2 sections, they together comprise 60% of your syllabus. So here you can just make an idea about how important this is, and accordingly you can start up with your preparation. So now let us see what is a marking scheme. As I already mentioned, there are 2 types of question. Either it is asked for 1 marks or 2 marks. So for every 1 mark question, you will get 1 marks for a positive and right answer. So if you are answering it wrongly, so there will be a penalty of -1/3 from the total marks. So you should be aware of this negative marking, and so is for 2 marks. For 2 marks question, there is a negative marking of -2/3 for each and every wrong answers. So there is no negative marking for MSQ and NAT. So you can just practice MSQ, you can clear your concept, and also you can practice numericals, because there are no negative marking for MSQs as well as NAT. So there is just a negative marking for multiple choice questions, where you are supposed to select a single option from the given 4 options. So yes, so now let's have more into the insights of the important topics of a syllabus one by one. So the first topic here was general aptitude. So what all topics are important under this? Coding and decoding, series formation, like what comes next, symmetry, directions, speed, distance and time, profit and loss. Sometimes the question on combinations and permutations are also asked, so you can focus on that part too. Now coming to the second portion, second section, that is chemistry. Again, this is a compulsory part for you all. Now here you can see there are a list of subtopics that you need to cover. So the first topic over here is atomic structure and periodicity. Under this, you should learn about Hund's rule and Pauli's rule. What is Hund and Pauli's exclusion principle? Also, you should know about the periodic table and the most accepted periodic table if it is given by Mondelez or Mendeleev. You should know what are the periodic property, what is atomic size, what is atomic number, how it is increasing across and down the periods and groups. What is ionization energy? What is the increasing and decreasing profile for electron affinity, electronegativity, and also atomic size? So you should just have a glance and practice numericals, you will get into it. Now, second topic over here is structure and bonding. Here you should know the basics of ionic and covalent bonding. What is the energy for this? What is VSEPR theory, that is valence shell electron pair repulsion theory. You should also know about the hybridization of a molecules as well as the shape. Also the structure parameters such as bond length, bond angle, as well as bond energy. Knowledge of these topics is needed. Now you should also know the basic property of S, P, D and F block elements. Under chemical equilibria, you should practice a numerical for elevation of boiling point and depression of freezing point. Every time you will find one numerical from this topic, so don't leave it. It's a suggestion. Just try to practice a numerical for either elevation of boiling point or depression of freezing point. Also, the knowledge of pH and buffer is needed. Under electrochemistry, you should practice EMF potential, how it is calculated, what is the formula for Nernst equation. Under reaction kinetics, order of reaction, what is first and second order reaction, what is activation energy, what is enzyme, what are their properties, how the inhibition of enzyme is taking place, what are the different types of inhibition and how it is regulated. At last is the chemistry of biomolecules, like we used to study in our biochemistry syllabus, like DNA, RNA, carbohydrate, proteins, lipids. What are their basic structures, functions, enzymes involved in their metabolic pathways and also their regulation. Now the next part over here is biochemistry. Core subjective part. Now we have come to part number 3, that is biochemistry. The first section is biochemistry. Here you should know about the biomolecules, again like carbohydrate, proteins, how they are synthesized, vitamins, what is the function of different fat and lipid soluble enzymes, vitamins, all these things you should study under biomolecules. What is protein structures? What are the different types of protein structure do we have? There are 4 types of protein structures: primary, secondary, tertiary and quaternary. What are the bonding involved in these structures? What is motifs? What is domain? What is the solubility of protein? How this protein degradation and denaturation is happening? What is enzyme? What are the different classes of enzyme? How it is activating the process? How it is increasing the rate of reaction? How the activation energy is changed with the help of enzyme? What are the functions of enzymes? What are the enzyme inhibition and also its kinetics, which include equation. What is the LB plot, that is Line-Weiberberg plot. Next, you should also focus on metabolism and bioenergetics. Here, glycolysis, TCA, oxidative phosphorylation, enzymes involved in these pathway, what are the inhibitors for these pathway, how this is regulated, all things. What is biochemical separation techniques? Make sure when I'm talking about techniques, it is always principles and function. What is the specificity of techniques? So what all techniques you need to focus here is electrophoresis, chromatography, spectroscopy, chromatographic, like how ion exchange chromatography is used. What is the specificity for affinity chromatography? Like all these simple topics. Also, plasma membrane. What is the structure of plasma membrane, which is the most accepted model? What are the functions of plasma membrane that it is taking care of? So for plasma membrane, just structure and functional aspect need to be clear. Basic processes like replication, translation, these are basic processes which we are studying since our bachelor's time. So you should know the basic mechanism of all these processes, what are the enzymes involved and how it is regulated. Under immunology, what is B cell, what is T cell, what is active and passive immunity, how the maturation of immune cells is taking place, what are the different cytokines that are secreted by this immune cells, how the recombination of B and T cell is happening, what is clonal expansion, what is monoclonal antibody production, what is the name of the antibiotic which is used to inhibit the de novo or salvage pathway in monoclonal antibody production, that is under hybridoma technology. So this is all about biochemistry. So let's proceed further. Now coming to botany. The first topic under botany is plant systematics, where you need to clear about the plant classification, its phylogenetics and cladistics. Also sometimes they ask about DNA barcoding. So these topics you should learn under plant systematics. Under plant anatomy, they ask about dicot and monocot root. If it is dicot root or stem or monocot root or stem, how it is appearing if we see under the microscope, whether collateral bundles are present in dicot root or monocot stem. Like this they ask. Under plant development, cell and tissue morphogenesis, this is a very important section as it includes a variety of topics like what is photoperiodism, what is vernalization, what are plant hormones, what is the secondary metabolite production, what is mineral nutrition, and how stomatal opening and closing is regulated in plants at the daytime as well as night time. Under plant physiology and biochemistry, again you need to focus on the secondary metabolite production the most as well as plant hormones. Sometimes they also ask about the bioenergetics and thermodynamics here. Genetics and genomics include the genome editing tool like CRISPR-Cas, what is miRNA silencing, what is the function of siRNA, how it is blocking the protein expression, gene expression, and also you should know about the basic concepts of genes like simple molecular biology is being asked here, like what is euchromatin and heterochromatin, what are DNA damages types, how this DNA damages is being repaired, what is transposon, simple genetics is being asked under botany. So what is economic and applied biology? Here a knowledge of economically and medicinally important plant is very necessary. Like sometimes they ask these questions in match the following forms. What is plant pathology? What are the nature of pathogens that are causing diseases in plant? What are the type of diseases that is caused by plant, that is caused by bacteria, fungi and viruses in plant? So let us take an example for this. Like if I consider an example of Phytophthora infestans, which is a pathogen that is causing a disease in plant. Which type of disease is this causing? Whether it is causing late blight of potato or early blight. So it is a question for you all. It's a homework for you all. So like this question, it is asked in exam. So you should practice questions on the basis of match the following form. And also the concept of ecology and environment is sometimes asked. Simple concept like what is niche, what is habitat, like all these things, what is the role of barnacles and how these barnacles are occupying the lower and the higher niche in their habitat. So the third section that we have under part 3 is microbiology. So here you need to know about what are the methods in microbiology, like what is pure culture technique, what is CFU, that is colony forming unit. Every year the questions on CFU calculation is being asked. They just give you numericals for this and you have to write the numerical value, like you have to mention the answer for this. So you should always focus on how to calculate this colony forming unit for a question. The next topic is microbial taxonomy and diversity. Here the importance and classification of bacterial, viral and fungal classification is needed, like what is the genome of bacteria, if it is gram positive or gram negative, what is double stranded RNA, what is single stranded double-stranded RNA, what is the genome of RNA virus, like all these things you should know. What is the difference between prokaryotic and eukaryotic cell? What is the difference in their structures as well as their functions? Functions of cell organelles that are found in the prokaryotic as well as eukaryotic cells. What is the difference in their glycerol backbone? If prokaryotic is more related to eukaryotes or archaeobacteria is more related to eukaryotes. Under microbial growth, you should know the 4 phases of microbial growth, that is lag phase, log phase, stationary phase, and then comes the decline phase. Also, they focus on the type of metabolites that are asked in these different kinds of phases. Is trophophase also called log phase? These type of questions are also asked. Stationary phase is also called as idiophase. So under microbial metabolism, you should focus on TCA, glycolysis, Krebs cycles, oxidative phosphorylation, their inhibitors, and also their metabolism. Along with this microbial growth topic, you should also focus on microbial growth kinetics. Sometimes a numerical is being asked. Sometimes instead of asking the numerical, they just ask about the formula. What is the formula for microbial growth kinetics? They will give you 4 options, and out of this you have to select one option. This is a suitable formula for calculating the microbial growth kinetics. So microbial diseases and host pathogen interaction, how this portion is being asked in MASH of the flying form. Like they give you the name of the pathogen and the related diseases that is causing disease in case of plants, animal, and human, because we are considering microbiology here. So pathogen which is causing diseases in any of the hosts such as plant, whether it is animal or it is human. What is chemotherapy? What are antibiotics? How it is acting? Whether it is inhibiting the cell wall formation of bacteria, whether it is inhibiting the protein synthesis of a pathogen, whether it is blocking the initiation or elongation pathway of a protein synthesis, that is translation. Just make a comparison chart for this and try to cover this portion. Now last comes over here is microbial genetics, which include the normal genetics, like what is transduction, what is transformation, who was known to perform the transformation experiment, how was it performed, what is conjugation, like all these things you will find in microbial genetics part. Now the next part over here is zoology. It mainly deals with the animal studies. So the first topic over here is evolution. Here you should focus on the theories of evolution. What is the historical perspective of evolution? How was the origin of a species? Theory was given for this. What are the factors or the ratio of gases that were used to prove the origin of a species? What are the different theories that are given by different scientists such as Darwin and Lamarck? What are the most accepted theory for evolution? Under genetics, again you need to focus on simple genetics, like what is pedigree. They will give you a pedigree representation, and there you have to determine the type of pedigree it is, if it is autosomal, if it is recessive. And also they ask about inheritance. What is cytoplasmic inheritance? What is maternal inheritance? Like this they ask. What is recombination? What is linkage? So these types of questions are asked under genetics. They also ask about common biochemistry that we have already covered earlier. Cell biology, like prokaryotic, eukaryotic structural differences, what are sorting mechanism, how is co-translation and post-translation sorting happening, what is the signal sequence for the different type of organelles transport. Under microbiology, you should know about the genes, difference between different euchromatin and heterochromatin, and all the basic processes like replication, transcription, translation, how it is regulated, what is alacoperon, how the CMP levels get varied in the presence of glucose and lactose, what else is asked about regulation, how these pathways are being regulated, how the ferritin regulation is happening. So all these things come under molecular biology. Under developmental biology, you need to focus on the very basic things like what is specification, what are the types of stem cells, what is induction, what is competence, what is determination, how the polarity in C. elegans is determined, how the polarity and the genes that are there that play a role in the formation of Drosophila body. Like they just ask in match the following question, genes that is responsible for the formation of development of polarity and axis in Drosophila, like maternal genes is there, parole genes is there, homeotic selector genes is there. So you can study like this way. Under animal anatomy and physiology, you should focus on the systems of human body like digestive system, respiratory system, chemical system that deals with endocrinology, reproductive system, excretory system, like all these basic functions and how this system does its own function. Gene expression in eukaryotes, how the gene expression in eukaryotes is regulated. And the last part over here is ecology, basic definitions and basic terminologies that are related to ecology is also covered under this sections. Now last section under part 3 is food technology. Here this subject we generally don't study, so I would suggest you to opt only the 2 choice of the 4 section that I have showed you in the previous slides. So if you want to opt food technology also, you can opt. So here what all things are important? Food chemistry and nutrition. Here you need to focus about general, again carbohydrate, protein, lipids, pigments, chemical and biochemical changes that are taking place in these molecules. Second is food microbiology, that focuses on the characteristics of microorganism, how the microbial growth is taking place, how the spoilage of meat, milk, all this is happening, how microorganism is causing the spoilage of these food, what are toxins, what are the different types of toxins that are secreted from microbes, whether it is exotoxins, endotoxins, food engineering. In food engineering, you have to study about heat and mass transfer principles, and also food products like how the milk is processed, what are the food standards and quality Wale rule like FSSAI, different kinds of standards are there, so you should learn this. What is HACCP protocol? Like this you have to focus for food technology part. So yes, so now let's study about the couples of tips and tricks for GATE Excel. What are the tips and tricks or hacks that are needed to clear and to study this GATE Life Sciences syllabus? So firstly, you have to prepare a strategic schedule plan. Firstly, according to your interest and according to your timetable, according to your routine schedule, you can just prepare a strategic schedule plan for you. Now, if you have prepared that plan, now it's the time to start or execute your preparation. For this, just read the reference books, because you're not only preparing yourself for exam. After clearing this GATE exam, you may have an opportunity of going for research or a PhD. So you should always clear your basic concepts for your future. So I would always suggest to read reference books for a topic. If you are reading while you're preparing for GATE, always try to make concise and precise note on a topic so you can refer it these notes for your preparation or revision purpose. And while you are preparing, also try to make a flashcard and a sticky note for it, because it is a way to manage and to divide the whole syllabus into simpler ideas. So always try to make flashcards, try to include mnemonics to make these flashcards more practical. And now if you are done with your study, it's a time to implement this, what all you have studied. For this, you can solve PYQs. Firstly, start with GATE. If you are done with last 10 years PYQs of GATE, then start with DBT. And now if you are done with DBT, then also start with CSIR. So if you are done with solving PYQs, now it's a time to practice mock test papers so as to see your preparation and also rate your success. Now if you're done with all these things, now it's a time to revise and recall. At the last, you should always revise and recall what you have learned. So firstly, study, practice, recall. You should always follow this SPR strategy for clearing any of the exam. So I would like to conclude my topic by saying just a single quote. It is not about how bad you want it. It is not about how bad you want it. It's about how hard you are willing to work. So thank you once again for watching this session. I hope you enjoyed this session. If you liked the session, do not forget to like, share and subscribe. Also share to your friends and colleagues whosoever is preparing for this upcoming GATE exam, so that they can also get a doorway to their success. So meet you in the next video. Till then, bye bye, take care, wish you all the best.